So once again, I welcome you all, madam. And madam is with us from the initiation of this lecture series. So thank you so much, madam. Now the platform is for you, madam.
Thank you
So
for joining, ma'am. You're most welcome. And as Achilles sir has said, ma'am is one of our member in this organization team. So it's your own stage. Welcome here, ma'am.
thank you so much. So today we are going to talk about fundamentals of research in Ayurveda, and especially we are going to talk about translational research, translational medicine, and evidence-based medicine. So for that, if Mohan sir permits me, I would like to invite my two colleagues first to give a brief presentation of their work, just to get an idea what translational research actually means and how we work. So if we can unmute Aishwarya Padhe Pense. So she will be presenting first for first 10 minutes and then followed by Dr. Sheetal Giramkar. So Dr. Aishwarya first and then Dr. Sheetal, and after that I will speak. So organizers, can you please unmute
Yes,
Aishwarya?
yes. Just a moment.
Sure.
कर दिया है.
Yes, good evening, sir. Good evening, ma'am. Good evening, everyone. Yes.
Good evening,
Am I-
ma'am.
I'll just share my presentation. Is my presentation visible?
Yes.
Okay. So, good evening, everyone. I am Dr. Aishwarya Padhe Pense and currently working as a postdoctoral fellow with Dr. Supriya. And I completed my doctoral research from Nanobioscience Group of Agarkar Research Institute, where my doctoral research topic was evaluation of zinc oxide nanoparticles in delaying the development of diabetic nephropathy. So, to begin with, we all know that diabetes is one of the metabolic disorder where there is elevated blood glucose, is hyperglycemia, and when diabetes is for prolonged duration, it is if it is uncontrolled and poorly managed, it show adverse effects on various organs of the body, various macrovascular and microvascular. So in case of macrovascular complications, the major arteries gets affected, stroke, diabetic cardiomyopathy, or affecting the extremities of the body. Whereas in case of complication, the small artery to defer damage, diabetic and diabetic nephropathy. So this diabetic nephropathy is one of the complications of diabetic mellitus associated with kidney damage. And the damage progresses to end-stage renal disease, which is a common cause of kidney failure among 45% of percentage of diabetic population. So at present, there is no specific treatment or cure for the disease, but it is managed through strict glycemic control and strict blood pressure control. And are suffering from end-stage renal disease have to rely on hemodialysis or renal replacement therapy for survival. So this is the gap that we thought we should find some way to prevent this disease. So previous work in our lab on Yashada Bhasma, particle of zinc oxide. So in our lab, we synthesized zinc oxide nanoparticles through chemical reaction and studied their anti-diabetic effects. And we found synthesized particle good anti-diabetic activity. There is in serum levels of as well as increased pancreatic cell mass in animal models. So carry out this forward, can zinc oxide nanoparticle treatment also delay the onset of diabetic nephropathy? And for this, we studied nanoparticle in as type 2 diabetic models, as well as in so how hyperglycemia causes this? Elevated glucose levels give great oxidative stress, which through inflammation, necroptosis, and renal fibrosis causes renal injury or kidney damage. This is the so starts by using a chemical that is pancreas.
Aishwarya, your voice is not clear at all. I mean, it is very breaking. Can you
Oh,
check your connection? We are not able to
yes.
hear you properly.
Okay. Ma'am, now is it clear?
It is clear now.
Okay. So we made Vistarad diabetes using a chemical that damages pancreas. It is named as streptozotocin. So after inducing diabetes in Vista rats, we administered zinc oxide nanoparticles orally for the period of 8 weeks with periodic measurement of creatinine, uric acid, and blood urea nitrogen levels. And at the end of treatment, the animals were euthanized and their kidneys were collected for histochemical analysis, gene expression studies to see what these zinc oxide nanoparticles are actually doing. So we observed that after treating the rats with zinc oxide nanoparticles, there was dose dependent decrease in serum creatinine, serum urea, and uric acid levels, whereas their excretion in urine got increased in dose dependent manner. So as we can see, this graph states the increased creatinine clearance as well as urea clearance from the zinc oxide nanoparticle treated rats. So as I said previously, hyperglycemia through reactive oxygen species gives rise to inflammation as well as fibrosis. So the genes involved in inflammation, that is NF-kappa B, and gene involved in fibrosis, that is TF beta, was analyzed, and diabetic rats had levels of both of these genes, whereas zinc oxide nanoparticle treatment significantly reduced the expression of these genes. And the histopathic pathological analysis by periodic acid shift staining revealed diabetic rats has damaged kidneys as compared to control healthy rats, and zinc oxide nanoparticle improved this structural damage or prevented this hyperglycemia associated structural damage. So the genes involved in this fibrosis and inflammatory pathways were also analyzed, and zinc oxide nanoparticle treatment improved their expression or suppressed the pathological genes involved in these pathways. So further, the effect was carried out. Podocytes are structurally specialized cells of the kidneys and in component of filtration barrier where the actual glomerular filtration, as we can see in this image, the entire yellow component is one podocyte cell. So it has a cell body, primary processes, food processes, and the food processes from adjacent podocyte from a molecule from actually blood filtration process takes place. So these podocytes have three important proteins, podocyte and W, and we studied the effect of nanoparticle treatment on these three proteins also. So loss of WT and podocyte results in loss of nephrin, which damages these podocytes and results in proteinuria, that is protein rich urine, which is one of the hallmark of kidney damage. So we determined the safe dose of these zinc oxide nanoparticles on podocytes by MTT assay, where we found that zinc oxide nanoparticles were safe up to 10 microgram ml. So taking this safe dose, we evaluated the anti-lipid peroxidation activity of these nanoparticles, and we found that podocytes treated with zinc oxide nanoparticles showed decreased extent of lipid peroxidation, improved mitochondrial membrane potential, overall indicating the use of zinc oxide nanoparticles in hyperglycemic condition improved mitochondrial potential, decreased the extent of peroxidation, prevent production as well as retain the expression of important proteins. So to conclude, delayed the onset of diabetic nephropathy
No voice.
as well as the prevent for slide, ma'am.
Hello?
Hello. Yes,
Uh,
ma'am. For conclusion slide.
no, there was no voice.
Can you hear me, ma'am, now?
Yes.
To conclude, zinc oxide nanoparticle treatment delayed the onset of diabetic nephropathy through inhibition of fibrotic and inflammatory pathway. As the hyperglycemia induced podocyte injury also. So in future, with Supriya ma'am, I'm going to study the effect of or the difference that is synthesized zinc oxide nanoparticles have different bioactivities, like how organophide or herbometallic formulation of this from synthesized zinc oxide nanoparticles, and also we are going to generate scientific evidence to basic Ayurveda concept uh, in future studies. So thank you.
So thank you, Aishwarya. Your voice was breaking in between, but still, I hope you were audible in between. So what I wanted to show and highlight here that Aishwarya has done extensive work on zinc nanoparticles. And this whole work was inspired on Ayurvedic concept or Ayurvedic formulation Jasad Bhasma, as she showed. So are we missing somewhere? Being an Ayurvedic fraternity, are we missing somewhere, and is this something which we should be doing as a translational research is one question for today's session. Now I request the organizers to unmute Dr. Sheetal Giramkar. She will be also presenting for 10 minutes and then we will start our actual presentation for the day.
It's done, ma'am.
Sheetal, you can start.
Today I am here to present my topic on evaluation of the effect of tripla on obesity associated
Visible.
cognitive impairment. Right now, ma'am, now it is visible?
Yes.
Okay. So this work has been carried out in Irsha Bharati Vidyapit deemed to be university. And first of all, I would like to thank Supriya ma'am to giving me this opportunity to present my work in this lecture series. So as we all know, obesity prevalence is increasing worldwide, and it is associated with different comorbid conditions like hyperglycemia or CVD, and obesity associated cognitive impairment is one of the important and highlighted area of research currently. So now let us understand how obesity influences cognitive impairment. In obesity, as we all know, saturated fatty acids, that is free fatty acids, are getting increased, and which leads to imbalance in antioxidant enzyme, catalysis in brain, and increase in the central and central brain leads to decrease in the cognitive function. So what are the postulated mechanism for this overall situation? That is like vascular damage to the CN.
Your voice is also not clear. Not clear. No voice. Hello, Sheetal, can you hear us?
Imbalanced antioxidant enzyme.
Yes, ma'am.
Hello,
Yes,
Sheetal.
ma'am. Yes, ma'am.
Your voice is completely breaking.
Now I'm audible, ma'am.
Yes.
Yes. Okay. So I'll
I'll
start
start
with
with
this
the
slide
slide
again.
again.
Echo, I think you have joined from two devices.
Yes, ma'am.
Yes, ma'am. Just a sec.
Now, ma'am.
Two voices.
Yes.
Two voice. Echo is there. Hello, Sheetal. You rectify your problem. Till that time, I will start. Hello.
Yes, ma'am.
Hello. Yeah. So you rectify your problem and you share at the you press.
Now my voice is audible, ma'am. Hello.
Yes, your voice is audible.
Okay. So shall I start with this slide?
Slides are not visible.
Now is it visible?
No. At least I cannot see. Sheetal. Yeah.
Yes, ma'am.
Now visible.
Okay. So I'm starting, ma'am. So let us understand how obesity influences cognitive impairment. As we all
Slide
know,
we
in obesity.
this slide was audible. Go to next slide.
Okay, ma'am. So on this background, we have hypothesized that AAA, which is a combination of three herbal drugs, that is Avla, Hirda, and Behda, you can be beneficial in this condition as it works in a weight loss due to its Meda Dhatu and Rasayana drug, and it is made up the formulation is made from the phalamajja of the fruits, middle part of the fruit, so that that can be beneficial in the action of brain or function of brain. So we hypothesize that tripla can be effective in obesity associated cognitive impairments. So though I have worked on four, five objectives, today I am presenting only three objectives over here, which mainly includes in vitro, in vivo, and in silico part of my study. So the objective was to evaluate the effect of tripla on free fatty acid induced neuronal lipotoxicity, where we have worked on SHSY neuroblastoma cell line. It is a neuronal cell line which was like available in the form of undifferentiated, but after using retinoic acid we can differentiate it into a mature neuron like structures. So we started with the proper culture and maintenance of the cells of differentiated cells, and firstly we have evaluated the cell viability with by treatment with trifla, where various concentrations here I have reported which are the concentrations we have used, that is 5 to 50 microgram per ml of concentrations. And in case of palmitic acid, to induce the lipotoxicity in the neuronal cells, we have used 500 to 2000 the well standardized on neuronal cells. And the second part was on effect of tripla on PA induced lipotoxicity. In both parameter reduced stress. We have evaluated lipid peroxidation, fatty acid profiling, where we have saturated more polyunsaturated fatty acid. Then neurotransmitters are playing main role in a cognition or brain functioning. So we have evaluated dopamine, serotonin, and noradrenaline level, and we have evaluated genes like antioxidant, specific genes with to antioxidant, neuronal plasticity, inflammation, and apoptosis, which are the main pathophysiological mechanism involved in the pathophysiological mechanism of obesity, lipotoxicity. So coming to the result part, here in case of differentiated cells, you can see the proper neuronal structures where the proper neuronal axons, neuroids are there. And after treatment with a palmitic acid, the neuronal morphology has lost. In case of cell viability, after treatment with a palmitic acid, which induces lipotoxicity in a cells, it was observed that the palmitic acid was cytotoxic from 1000 micromole concentration onwards, and when cells treated with the palmitic acid only and along with a safe concentrations of AAA, that is 10 and 15 microgram per ml, it was observed that AAA's highest concentration shows rise in the cell viability. Coming to the next oxidative stress marker, lipid peroxidation, as brain is rich in the lipids. Here, the after treatment with a palmitic acid or after causing the lipotoxicity, there was increase in the lipid peroxidation, and AAA treatment group arrested the lipid peroxidation. Then coming to the fatty acid profiling, here it was observed that when cells were treated with a palmitic acid, there was the fatty acid levels was got altered, where you can see saturated fatty acids were has got increased as compared to control cells, and it was found to be decreased in higher concentration of AAA treatment. And coming to the monoamines, that is the neurotransmitters, the here in all monoamines, palmitic acid treatment resulted in a decrease in the levels of the dopamine, serotonin, and noradrenaline, but after treatment it has shown a dose dependent increased levels in the monoamines. Coming to the gene expression, where we have selected NF2L2 as an oxidative stress marker, where it was observed that it has got activated under oxidative stress, that is palmitic acid causes oxidative stress, and AAA treatment resulted in a decrease or a down regulation as compared to palmitic acid. In case of BDNF, brain derived neurotrophic factor, it is a neuronal plasticity marker. It was observed that as compared to palmitic acid, AAA treatment group showed upregulation of the expression in dose dependent way. So it may increase the neuronal plasticity. In case of inflammation, TNF alpha, the inflammatory response upregulated by but did not change by the AAA treatment. And FAS, which is an apoptosis marker, where it was PA induces apoptosis in all the traits on the all the groups, and there was a down regulation in AAA's highest concentrations. Just to note that the experiments were carried along for example, like in control cells were not treated with anything, like they are the normal neuronal cells. In case of palmitic acid, it is treated with only palmitic acid, and in case of treatment group, the palmitic acid along with AAA was given to the cells. So coming to the in vivo study, it was effect of AAA in experimental model of cognitive impairment associated with high-fat diet induced obesity. So this experiment was carried out in two parts. Firstly, we have carried out first one in young rats, where the high-fat diet was given to the rats for completely 120 days, and the AAA was given for the 30 days. So this experiment, as per the rules or guidelines, it was started with the institutional ethics committee approval, and the groups were like disease control, positive control, which was rivastigmine, then AAA's low, middle, and high dose. And second part, where we have changed our parameters of experimental design, where we have given the treatment to the animals for 60 days, and high fat diet was given as that of part 1, that is for 120 days. Here also the two doses, which was like middle dose of earlier experiment and higher dose of middle dose was taken for the experimental conditions, and disease control only high fat diet was given. In case of positive control, we have used atorvastatin. Assessment variables were evaluated during this experiment was body weight, serum profile, serum markers like lipid profile, fasting blood glucose on particular days like day 0, 90, 120 in both the parts. Then IPGTT, intraperitoneal glucose tolerance test, and insulin levels. Then we have assessed behavioral assessment, behavioral parameters as it was focused on cognitive functioning. So Morris Water maze and elevated plus maze parameters were evaluated for their short-term and long-term memory. Then brain homogeneity was evaluated for lipid peroxidation marker as that of in vitro experiment, acetylcholinesterase activity, monoamines level as that of in vitro like dopamine, serotonin, and norepinephrine, fatty acid profiling, and gene expression studies for NF12 to BDNF, TNF alpha, and FAST. I would like to mention that we have kept the parameters same as that of most of the as that of in vitro cells, in vitro experiment. Then coming to the result, here we can see that there was like a weight gain pattern for the animals during the experiment, and rivastigmine, which is a positive control, has shown decrease in the weight, but not in the tripla treated group as that of. In case of IPGTT, intraperitoneal glucose tolerance test, the tripola's highest dose has shown uh, clearance of glucose as compared to disease control and other treatment group. And in case of lipid profile, there was significant change in all parameters as compared to day 0. And in case of high fat diet group, there was a further imbalance in all the parameters as compared to day 90. Whereas on day 120, AAA showed significant decrease in cholesterol level as compared to HFDC group in case of highest dose. Coming to the cognitive performance, where Morris Water maze was performed for short-term memory. Here, animals are trained for finding the location of a platform to rescue themselves from the water. So here we can see it was a visuospatial learning and reference memory performance. It was carried out. It was evaluated on day 90, that is before treatment, which is known as acquisition latency, and on day 120, which is retrieval latency, like how they retrieve the platform location. So here it was observed that escape latency time for AAA treatment group 1 and AAA treatment group 2 showed decrease in time to find this platform, which shows that it increases their learning or short-term memory. Then coming to the expression of selected genes, where it was observed that NF2L2, which is the oxidative stress marker, was found to be upregulated by AAA treatment, and there was no significant change in case of BDNF or inflammatory TNF alpha and apoptosis fast gene. So other parameters has showed slight reduction after repeat treatment, and then as I have already mentioned in part 2, after conducting part 1 experiment, the experiment we have performed again, and where we have changed our methodology or study design by intervening the tripla on more days, like for 60 days. So instead of going in detailed result, I have made a comparative table for it. So in case of part 1, where we have given tripla treatment for 30 days, there was no change in body weight, but after giving the 60 days treatment, there was significant decrease in the body weight pattern. Then lipid profile and fasting blood glucose, significant change was observed only in cholesterol levels. But after increasing the treatment duration, there was a significant change in all the parameters. Similarly, in case of lipid peroxidation, there was a decreased level of lipid peroxidation significantly after more days of treatment. Acetylcholinesterase activity was found slightly decreased after increasing the treatment. Similarly, in case of brain monoamines, there was increase in the levels in a treatment groups for all the monoamines. In case of saturated fatty acid, and or in case of fatty acid profiling, there was a slight increase in the N3-PUFA, which is essential fatty acids. And in case of gene expression pattern, there was no change in the expression levels. So after this, to evaluate the mechanistics, we have conducted in silico study by selecting the 7 bioactive compounds, that is ethyl gallate, ellagic acid, epicatechin. gallic acid, quercetin, ascorbic acid, and shebulinic acid. These 7 compounds are common from all these individual drugs, that is Avla, Hirda, and Beda, and they were further evaluated for their docking studies for with respect to obesity associated cognitive impairment and the specific gene selected for this postulated mechanism like oxidative stress, inflammation, neuronal plasticity, and apoptosis. So further this we have carried out ADMET analysis and toxicity studies where we found all drugs are safe, all the compounds were safe, and the further was protein ligand interaction were also carried out to how it is working. So where I am showing here only the ligand ligand protein ligand interaction where out of these 7 bioactives, ellagic acid was found to be more effective or more protein like we can say affinity binding affinity was more for ellagic acid for inflammation, apoptosis, oxidative stress, and in case of BDNF, though quercetin was there, but along with quercetin, ellagic acid was more potent for all these things. So to conclude, or what is the take-home message? Like Triphala has shown neuroprotective effect by modulating monoamines and N3-PUFA levels in both part of the studies. These findings need to be confirmed in further dose and duration finding studies, like by standardizing the model, like study drug intervention, where we have increased the dose of duration of the this thing Triphala. Then chronicity of study model also needs to standardize. Like in case of part 1 study, there was a long-term what we can say high-fat diet was given to the animals, but after increasing the dose duration of tripla in second part for 60 days, the high fat diet was given for only 60 days to the animal. Instead of first part, it was for 90 days. Then using a advanced system and the mechanisms in detail, where it will help to test the hypothesis in proper way. Then to screen the candidate molecules, like we have evaluated 7 bioactives, and to analyze the complex system more efficiently and cost effectively, we can take help of in silico studies too. So this was the overall presentation. Thank you, ma'am. Thank you all.
So, thank you, Sheetal. So, in this presentation, what we learned that we started with Triphala and why we use Triphala for obesity-induced cognitive impairment, because though it is not very clearly written anywhere in the Ayurvedic text, we thought that since we are using Phala Majja of 3 drugs, maybe it is useful in Majja-related diseases. So, we thought that, and since Triphala is anti-obesity, I mean, it is known for its anti-obesity effect. We thought that can we merge these two things? And that is how we evaluated in obesity-associated cognitive impairment. The interesting part which Sheetal showed and we never work upon is like if there is a longer duration of obesity, then we have to give treatment also for longer duration, and that we have proved in this study that when she gave treatment only for 30 days, it didn't work. But when we gave treatment for 60 days, it worked. We used in silico studies, which you have learned in the lecture on 1st October, but we did not only did the in silico studies, we just tried to complement our studies, our finding that if somebody asks us, because this is quite often asked question that which molecule of Triphala is working. Though I am not interested in going into dissecting the Triphala and finding out the molecule from that, but this was important for us to answer this question that which of the 100 molecules which are present in Triphala, which is more effective in this particular condition, and we found that ellagic acid is the most potent molecule. We have observed one more thing which Sheetal has not showed. The 7 molecules which she has identified, none of the molecule crossed blood-brain barrier. We have learned what is blood-brain barrier in one of the lectures. So if we have to cross, I mean, if we have to make Triphala to cross blood-brain barrier, we have to rely on Ayurvedic concepts like making grutha of Triphala and not only the Triphala extract or Triphala kashai. We have to use in this condition so that Triphala can pass the blood-brain barrier. So these are some lessons or these are some things which we can take, and this is actually we can call as translational research. Now I coming to the my own topic, which was given to me, and I'm going to quickly run through the slides, because in my presentation we are not going to learn any new technique, but there are few new terms probably which we will quickly run through. So this was the topic which was given to me, and this was the agenda, like I have to discuss the importance, areas and challenges of translational research in Ayurveda. And for that, it was very important to learn what is translational research. We will see few more examples also. Translational medicine, and then evidence-based medicine and its framework. So these are three broad topics which we are going to cover. Now, let us consider a big picture that most discoveries never reach practice. This is not about Ayurveda, but in general also, most research never reaches the practice, never goes into clinics. And you will always find, in fact, in Ayurveda, this phenomenon is very prominent, that practitioners will say like, no, we don't know anything about research and we don't understand anything. So they keep themselves away from research. And researchers also do not take efforts to reach to practitioners and then convert our findings. Imagine that in Ayurveda, almost 10,000, more than 10,000, I don't know exact number, but 1956 was the year when Ayurveda PG has started. From 1956 till 2025, how many theses have been published in Ayurveda? Just imagine a number. And how many findings of those theses have literally gone into the clinics? In thesis, everybody writes that my drug is excellent, my thesis is excellent, whatever I have done is excellent. Then where this excellence goes? Where are the drugs? Where are the concepts getting translated into the practice? Translated in the sense used in the practice. You may say like, we publish. Some of us now publish. But is publishing sufficient? That is also a question. Nowadays, if you go for higher positions, higher institutes, they don't just ask you like how many publications you have, but they ask you what is impact of your publications. And what is in publications impact? If you see, there are different kind of publications impact. First is academic impact, and every one of us knows this very well, that we should have citations, we should have good H index, there should be some inclusion of our articles in systematic reviews and meta-analysis. So this is all academic impact. But more importantly, there has to be some policy impact. That means research cited, our research should get cited in some government guidelines, expert committee reports, legislations. Then it is called as policy impact. Methodological or technical impact, where tools are developed, questionnaires are published, people are using those questionnaires, protocols or guidelines. This is methodological or technical impact. Then there is a translational or clinical impact, where the evidence move into practice. Actually, physicians start using that evidence. Whatever I have done, if I'm saying that Triphala is useful in obesity-induced cognitive impairment, how many clinicians have started using Triphala for cognitive impairment in obesity? That is something which has to be measured. New products, improved interventions, or some clinical trials informing the treatment. This is a translational or clinical impact. Then commercial or industry impact. You might have heard a term like technology transfer, data transfer. So this is related to commercial or industry impact, where the work supports some product development, licensing, patents, pharma collaborations. Then there is educational or capacity building impact. That means based on my article, suppose some training programs are arranged, some curricular reforms happen, new teaching modules are developed, then this is called as educational or capacity building impact. And lastly, there is societal or public engagement impact. That means the articles get covered in media, newspaper features, they become popular articles. Somebody makes your research article as a popular article, then it is referred in public talks, or someone invites you to give a public talk about your hi-fi research, and ultimately it should improve awareness. Suppose I am working in pre-diabetes, then there should be increased awareness about pre-diabetes in public. Then I will say that there has some public engagement impact of my work or my research or publications. So in short, we should go beyond the publication and bridge the gap between lab, clinic, community, and policy. And there is therefore need for translation, because whatever we do now, when I say lab, it just doesn't mean that lab means some animal study or cell line studies. Whatever work we are doing in research, as our MD thesis, PhD thesis, faculty projects, whatever work we are doing, that work should get translated or should be used in clinics, should go to community, and or somewhere it should reach to policy. Now, of course, you will say that everyone cannot reach to policy, but at least our research should have some kind of impact, that it should at least reach to clinics. Now, you will say that we all are Samhita Siddhanta students or faculty. So how our concepts or whatever we are dealing, it can be translated. So just take two examples. One is concept of Agni. So lab or bench, lab work, it is also called as bench work. So what we can do in lab work, that identify some biomarkers for digestion and metabolism. So we have heard about gut microbiota. Can we have some signatures like gut microbiota? In Mandagni, this is gut microbiota. In Vishamagni, this is microbial patterns. Then digestive enzyme activity that we can measure, like in Mandagni, what happens, in Vishamagni, in Tikshnagni. If we want to go further, we can do metabolomics. Now, again, one thing is there, and people ask, like, we are working in Ayurveda college. We do not have these facilities. How we can do this research? Who will fund us? So remember that every work doesn't need high-fi funding also. If you have a concept and if you are able to convince it appropriately, properly to the opposite person who is a scientist working in some national institute, central institute, then that person may help you, because if he finds your concept interesting, if he says that there is some potential of getting translation, then he will definitely help you in doing your projects. Please let me tell you that even my lab doesn't have very high funding, or I am not like in a national institute. We are working in a private university, so we also do not get huge fundings. But the work of gut microbiota, which Dr. Sarika referred to you, that we have done together. So I have approached a scientist who is working in NCCS, that is a Institute National Center for Cell Sciences, is a DBT Institute, and there is a microbial culture lab. The whole facility is there. I approached that sir with my work on Triphala, that I wanted to study effect of Triphala on gut microbiome. But he was like that, this big project, whenever you will plan, it will come. But let us start doing something. And then we both came to a conclusion that we should start with buttermilk enema. And that is how we did the study. We did the patient part, and the stool samples were sent to him, and he did the whole analysis free of cost. It is your ability how you convey your idea, your concepts to the opposite person to get help for your work. So we have discussed like beige. Then what happens at bedside? Correlate Agni assessment with metabolic disorders. For example, if there is a Sthaulya patient, Prameha patient, Grahani patient, what is the status of Agni and what is the status vis-a-vis these biomarkers? Then in community, you can develop Agni strengthening diet, lifestyle interventions, community education modules that don't eat if you are not hungry, don't eat between 6 hours or something like that. You can make modules and take them to community. And lastly, policy, it is guidelines for preventive health checkups, workplace wellness modules. These can be developed and this can be implemented at, say for example, some IT institute. So these can very well go in at least that workplace policy. So this is how you can work on the concept of Agni. Another concept is Prakriti. We all have participated in Desh Ka Prakriti Abhiyan. So what is like how we can translate this Prakriti? So first is bench or lab. In that you can develop genomic, epigenetic, microbiome correlates of Prakriti types. Bedside, we can stratify the patients for personalized therapy. For example, predicting drug response. Let me tell you, there are no studies, or if I am wrong at this time point, there will be hardly any studies which are based on predicting drug response as per Prakriti. So far, we have just done survey like which Prakriti is common in which disease, but we have no studies to show that how Prakriti predicts the drug response. Genomic signatures, yes, like CYP2 patterns differ, HLA1 some patterns differ, but actual that given the drug, how the response differs. Disease risk, we have no study that from childhood, if we follow some 10 Kapha Prakriti, 10 Vata Prakriti, 10 Pitta Prakriti individuals, and then see in later age that which disease condition they like are prone to. There are no studies. Community, if we say community translation, then tailored lifestyle and dietary recommendations at workplace, school, community level. And if we want to get this Prakriti into policy, precision public health models, insurance packages, workplace health guidelines, these are the policy things which can happen. Again, I am saying not everyone will be able to reach to this policy, but there has to be some effort starting from bench to move to policy side. So, coming to the translational research, what is it? It is a broad scientific approach that seeks to translate discoveries from basic science, that is bench work, to practical application. These practical applications can be anything: bedside, community, or policy. So, the focus of translational research is moving knowledge across stages. Like you will not just keep it at one stage, but you will move the knowledge from one stage to another. And there are 4 stages which are roughly identified. The T0 or T1 is a basic science. This is a first in human studies. You do a well systematic studies. T2, that is the next stage, is clinical efficacy. And from there, we have to move to clinical effectiveness. Now, these two words, efficacy and effectiveness, are different, and you must have learned that in your research syllabus. T3 is taking this to clinical practice and implementation in healthcare systems. And lastly, as we have seen, public health impact. So it can be achieved only through policy guidelines and community interventions. Now, let us take example of Triphala in obesity. If we have to go through these all stages, then the T0, that will be a basic stage, which will be just conceptualization. That you will just have some hypothesis, frame scientific hypothesis from Ayurvedic concept. T1 will be preclinical studies, and there you can study some in vitro or in vivo. I have highlighted these few, and why that I will tell you in some time. T2 is early clinical studies where you will have safety and initial efficacy. Then T3 is clinical efficacy or effectiveness, which is in RCT in both Ayurveda and modern clinics. And lastly, T4 is community and policy. So why I have taken this example? Because I work extensively in obesity and metabolic diseases, and we also work on Triphala. Now keep this template in your mind, and I will show like how we have traveled. Now this is a T0, that is hypothesis forming stage. So we have done a marketed Ayurvedic herbal or anti-obesity product survey, and we showed that it is the formulation. A Triphala is an ingredient almost in all formulations which are available in market as anti-obesity, herbal or Ayurvedic, both. Then we did utilization of Triphala by Ayurvedic physicians for management of obesity. So how many Ayurvedic physicians use Triphala for management of obesity? We also did a study in community to show that what are the knowledge, attitudes, and practice of Triphala in the community. So we came to know through this survey. This survey was done in 4 different parts of the country. You can see from the names of the authors also: Anupama Krishnan from South, then Sheikh Sikha Lekhru from Northeast, Vaishali Mali is from Chaudhary Brahma Prakash Samhita department again. So this was a pan-India study where we screened 1,000 individuals, almost 806. It is the number you can read here. And we found that Triphala is known to all parts of the country. People know it. People know that it is a safe formulation. People know that it is used for, not for obesity, but they were knowing that it can be used as laxative. So reasonably good knowledge was there in the community, which helped us to take our drug further. Then we did T1. We moved to T1, and then we showed that how Triphala regulates adipogenesis through modulation of expression of adipogenic genes in 3T3L1 cell line. And we also showed that how it works on molecular and biochemical level in high-fat diet-induced obese rats. So this forms a T1, that is the stage 1. From here, we went ahead and we did a preliminary clinical evaluation. So it was efficacy of selected herbal formulations on anthropometry and body composition in overweight and obese individuals. So this paper is also published. I again want to tell you something here. Many a times we feel that, okay, madam is doing something because she is in this institute, she has facilities. In this particular study, there is no biochemical parameter also used. Not even lipid profile we have done. It is just doing anthropometry and body composition. And this journal, Herbal Medicine, is its impact factor is 3. And you can publish a good paper if you like you have the information properly presented, then your paper without any biochemistry can also get published in a good journal. So this was our T2. Moving ahead, now we are at T3. You can see here that this trial is registered very recently on 28th April, and here we are going to do a randomized parallel group study and along with standard of care. So one group will receive Triphala and another group Triphala and lifestyle, and other group will receive only lifestyle, and this will be comparison. And this study is funded now by ICMR. So we are now able to do many investigations in this study. We are going to do even the DEXA scan of the individuals, and this is a multicentric study which is going to be done in around 400 individuals. So now we have moved from T0 to T3. Now you will say, like, what is a big deal in Triphala and obesity? It is well known. But let me tell you that when we started 10 years back, this whole work is of 10 years. When we started in 2014, there was not a single study published on Triphala from India. There was only one clinical study available, and that too was from Iran. So it was like a surprising thing for us that the formulation, which is so common and used in Ayurveda, there was no clinical study from India. So now we are moving to T3, and now next stage will be T4, where we have to take this to policy. But that is not my cup of tea, and we have to collaborate with institutions like All India Institute of Ayurveda, because to reach to that level, we need some central or national institute backup. I would tell you one more story. This is my mentor who was working on Rasayana, and she had a hypothesis T0. How Rasayana works. So they stimulate immune cells which set cascade of event, and that is how multiple actions are exerted. Now she is my mentor, so she could reach up to T4, and I could show you that, like, how she reached to T4. She selected Tinospora cordifolia, and she had her own reasoning why Tinospora cordifolia or Gulvel or Gudici from all the Rasayana drugs, because it is recommended for multiple indications used irrespective of Prakriti, so on and so forth. Then she did extensive studies using in vitro and in vivo models, and every cell which is involved in immune system or immunity, polymorphonuclear cells, macrophages, monocytes, reticular endothelial system, they showed. And you can see here this work is done in 1999, when the word translational research was not at all there. So this is a T1, in vitro and in vivo studies. And then they did lot of clinical studies. For 20 years, every student who ever used to come to Dhanukkar madam for doing MD or PhD has worked only on Tinospora cordifolia. And ultimately, this resulted into T4, that is a scientifically proven natural immunostimulant they could brought in a market, and that was not marketed by an Ayurveda company but an allopathy company. So it was real translation, because when we say clinics, so it should be Ayurveda clinics or allopathy clinics. Here they could reach even up to even to the modern medicine clinics, and this Immunomode is still available in the market. So from 1999 it is in market, and this is how stem to peel, that is how it was called, the story of stem to peel. So this is actually a translational research. Now, what is importance of translational research in Ayurveda? It bridges gap between the tradition and modern science. Very obvious. I don't have to read this. It creates evidence. It opens a innovation pipeline, because from there you can like have many things. You can convert Ayurvedic concepts, formulations, and practices into validated drugs, nutraceuticals, diagnostics, lifestyle modules. About this lifestyle module, I just want to tell you, if you go to Google app and just type Dinacharya, you will find there are n number of apps available. This is actually the commercialization of Ayurvedic concept. Now, whether it is backed up by Ayurveda concepts actually or not, that is another question. But what I want to show you, or what I want to tell you, that this is how we can commercialize also our products, and this is actually translational research. Health equity and accessibility. So it provides affordable and culturally congruent solutions. So when we have thought of working on Dinacharya, we have said this word, culturally congruent, because if you go to apps which are like not actually Indian or which are based on Western philosophy, their whatever the questions they will ask in their daily regimen are much different than what we will think from Ayurveda perspective. So culturally congruent or culturally very close to us, these kind of solutions we can offer, and ultimately policy impact is something which we all want. So what are the key areas? We have already discussed many areas like conceptual translation, drug or formulation development. I am not going to talk much about it, because being Samhita or Siddhanta person, I don't think we have to discuss as of now this particular thing here. But Triphala and Rasayana examples were from drug or formulation development only. Diagnostics and personalized medicine based on Prakriti concept, or having some Ayurvedic diagnostic frameworks. Why can't we have something based on Dashavidha Pariksha? So that is a Ayurvedic diagnostic framework which will help us. So can we have AI based module for the Dashavidha Pariksha? Lifestyle and preventive modules. So here we can develop some testable, implementable health modules which can be used for workplace well-being, stress management, cognitive enhancement, n number of things. Public health and implementation science. So applying Ayurveda-based interventions at community level for health system integration and cost effectiveness studies. So these are the different key areas of translational research. What are the challenges? We all know that there are scientific challenges, because there is lack of well-defined biomarkers for Agni, Amo, Ojas. We have to develop these markers. Limited crosstalk between Ayurvedic experts and modern lab or clinical scientists. We somewhere hesitate, and even if I have seen like there are many young students, so I would like to tell that generally when we approach anybody and if we start saying, you know, there are Dashavidha Pariksha, then Prakriti, Vikruti, Sar, Samhanan, then Prakriti is this. So that modern lab person or clinical scientist is not going to understand your Sanskrit words and your jargon of shlokas. So think like how you will present your ideas. Methodological challenges. So designing clinical trials that respect Ayurveda's individualized approach is very, very crucial thing and not so easy task. Lack of validated outcome measures that capture both Ayurveda and modern endpoints, because I many a times say like even the clinical trials which we do at our place, we use the modern biomarkers or modern endpoints. Why can't we have some Ayurveda endpoints? So then the question comes that there are not validated outcome measures because there are not no tools available. Suppose I have to go for Samyak Vamana Lakshana, then I have to always develop my own tool. It is not validated, then I use that only. So it doesn't make much sense. So we do not have many validated outcome measures available. Then we have regulatory and infrastructural challenges also. So if now I have to take this Triphala ahead, should I go as a modern medicine? We all know that Ozempic has done havoc in the society. If I have to stand with Triphala against Ozempic, how should I go about? What route should I like take? Drug, nutraceutical, functional food? What is the solution, or what is the way forward? Limited translational research funding for Ayurveda compared to biomedicine. Whenever we send proposals to DST, DBT or ICMR. First time this proposal of Triphala got funded by ICMR. But otherwise there is only one line written that it is Ayurveda project, go to Ayush Ministry. But now things will move, change slowly, because doing research on Ayurveda is not only our responsibility, but it is nation's responsibility. Fragmented research infrastructure and inadequate longitudinal databases. If I ask you, what was the Prakruti distribution in Goa in 1925 and what is the Prakruti distribution now in 2025, is there any change in the pattern? We have no data. So whatever research we have is not scattered, it is scattered, and we do not have longitudinal these databases that what was in 1925, what happened in 1975, now what is a 2025 situation. So these are few things. Cultural or perceptual challenges. Whenever we talk about Ayurveda research, there is always a skepticism that whether it will work. Zinc nanoparticles are acceptable, but Jasad Bhasma is not. So this is like something there, and that's why the translational research has some hurdles. Tendency to either oversimplify Ayurveda into single molecules or over romanticize tradition without rigorous validation. So there are two spectrums, either oversimplify or you jump onto anything without any validation. So both these things are harmful and we have to be balanced and at neutral stage. Moving forward from translational research to translational medicine. So what is translational medicine? It is just one slide. When you talk specifically about clinical implications of research, it becomes translational medicine. So we have seen that translational research goes into policies, it goes into communities, it goes into clinics, but translational medicine has a limited scope that if you convert it into clinics, then it is a translational medicine. It is more practice oriented, but it emphasizes multidisciplinary team because you want to convert something, some research into clinics. So clinicians have to be there, pharmacologists to know the drug in and out. So here pharmacologists, I don't mean that modern pharmacologist only. Ayurveda, Dravya Gun experts are also pharmacologists and they can also serve role here. And public health experts. So here Swasthavritta people also can take part, that how we can take the research into the clinics. So this is translational medicine, which is a subdomain of or subset of the translational research. Now, the last part of the presentation is evidence-based medicine. So what is EBM? That is how it is called as. So EBM is like a clinical practice, philosophy and method which integrates the best available evidence from clinical research, clinical expertise and patient values or preferences in decision making. Now this decision is for what? Which medicine to be given to a patient. So that decision is based on three factors: best available evidence, clinical expertise and patient values. So it is a decision making framework than the discovery oriented. So here there is no research, but it is like how you make decisions. So what is example of this is there are 10 antihypertensive drugs available, which you will select for a patient who is sitting in front of you. So you will refer some systematic reviews, maybe clinical guidelines which are available, and the patient's comorbidities, like what is patient. If he is diabetic and hypertensive, then your drug may differ. If he is obese and hypertensive, your drug may differ. Young hypertensive drug may be different. So patient's condition and preferences. So it is a framework which combines the evidence plus expertise plus values. So these three together will form evidence-based medicine. So when put into actions, the principles and methods of EBM ensure that medical decisions, guidelines and policies are based on the current best evidence about the effects of different forms of treatment and healthcare in general. So evidence is the main pillar of it, but clinical expertise and patient preferences also taken into consideration when we talk about evidence-based. With respect to medicines, it draws heavily on information from the benefit and risk evaluation. So which medicine is beneficial? How many risks and how many benefits are there from giving a specific medication to a patient? Why clinical experience is important? Why can't we go just by the evidence? Now there is so much of evidence, so why not to go only by evidence? Why clinical experience? Because the clinical experience is from the long standing practice of the clinician. He has some findings, he has some observation. It is also called as anecdotal evidence sometimes. So that's why this clinical experience is very, very important. And the role of clinical experience in the decision making process is very crucial, because without clinical experience, the risk related to a given treatment may end up causing unwanted effects. Whenever we plan any study in our department, we have a discussion with clinicians. Always this is a practice that I will call someone uh, who is practicing in that specific area, and I will ask the inputs of that person, that whether I'm going in the right direction. Should I improve my drug choice? Should I improve my clinical conditions, inclusion exclusion criteria? So even in research we give importance to this clinical experience. So clinical experience is of paramount importance. Patient engagement, because we saw that patient values are also important. So why patient, what patient will do when you talk about EBM? He will read, understand and act on the health information, and he will work together with clinicians to evaluate and select the right treatment options and providing feedback on outcomes. So he will not just like help in selecting, okay, I don't want woman, I don't think that I would like to have woman. This, if your patient says, you cannot force, because this is patient value or patient preference. Patients can have an active role to play at all levels of evidence. And it is important that patients have knowledge of EBM, that we are like implementing EBM, because it enables them to make more informed decisions. It also gives patients a more accurate perception of risk, because you are going to tell the patient that there are two treatments. If you go by one, this treatment, this will be the advantage. If you go by this treatment, this will be the drawback. So now I feel that this is better treatment for you, but if you have anything to say, you please tell me. So this is like you will make the patient in the process of decision making. Now this EBM is a 5-step model. It is very simple. Assess the patient condition, ask a relevant question, then acquire the best evidence, and then the fourth one is appraise the evidence, and lastly apply the evidence. So in short, assess the patient is history, physical examination, lab investigation, differential diagnosis. Through this you will assess the patient's condition. Asking a question is very interesting. Here also that PICO format is useful, which you use in framing research question. That same PICO format is useful in EBM also. Now, how it is important here, or how it is useful here? A patient is sitting in front of you. So P is the patient who is sitting in front of you, considering all his characteristics, that he is old, he is having diabetes, he is having hypertension also, he has history of MI in the past, whatever it may be. This is patient. Intervention is like you want to give best possible treatment to this patient who is sitting in front of you. C is control, that is routinely used intervention. So you can just give one intervention which is generally given in this condition, that is your control. But you want to find out, is there anything above this? And O is desired outcome. O is outcome. So desired outcome for that patient, whether you want to expand the lifespan of the patient, whether you want to give him some kind of pain-free period, what is your outcome? So that you have to consider here. And then you have to acquire the best evidence. So there are the levels of evidence. I am sure everyone knows this, and I am going to, I mean, these slides I am going to even post in the Google Classroom, and I am sure you all knows these levels of evidence, like from starting from the opinions to case series to case reports to case control studies, cohort studies, RCTs, and lastly meta-analysis. So all these characteristics have been mentioned here, and I will post these slides in the Google Classroom, so you can read if you want afterwards. The fourth step is appraise the evidence. So what you would do here, like whatever evidence is there, you will ask what are the results. Whenever you read any paper, you will ask what are the results, whether the results are valid. So how will you come to know whether the results are valid or not? You will check the methodology also. and you will try to find out whether these are genuine or not genuine. And lastly, you will see like, are the results suited for the patient who is sitting in front of you? There may be anything written in the results and discussion part, but whether it is like suitable to the patient who is sitting next to you, that is something you will think about. And then you will screen the evidence for internal validity and relevance. Okay, this is a paper of hypertension, but this is not suitable for me because it is not talking about hypertension in diabetic patients. You will not consider because there is no relevance. So this is how you will appraise the evidence. And lastly, you will apply the evidence where the role of patient values preferences comes in. So you will consider that whether patient is financially that like sound that he will be able to go for this treatment. My woman package is of 7,000 rupees, but whether the patient sitting in front of you is able to give this 7,000 rupees. Contraindications and allergies. So whether the patient is having any of these things for the medicine which you are prescribing. Preferred dosage forms. Oh no, I don't want ghee. I have aversion to ghee. I don't want any kind of ghee. So you have to respect because this is a patient value or patient's preference. And then considering all these things, integrate the evidence with clinical experience and patient values. And then when you give any treatment, assess if the intended outcome is achieved, whether the pain is relieved, whether the blood pressure has gone down, whatever is your outcome, and adjust treatment decisions accordingly if needed. So what is the benefits of EBM? It improves the quality of healthcare. It improves the patient satisfaction because he's part of it. So he doesn't feel like डॉक्टर ने मुझे दे दिया ऐसे ही, मुझे चाहिए था या नहीं ये पता ही नहीं है, इतनी महंगी ट्रीटमेंट मेरे लिए एप्लीकेबल थी या नहीं, डॉक्टर ने ऐसे ही पैसा कमाने के लिए दे दिया. This kind of unsatisfactory responses will never come. It will reduce the cost, and it is for self improvement of a physician also because he will read the evidence, he will get updated with the research which is being done in the field. So these are the benefits of EBM. So last slide, what is a comparison of translational research, medicine and EBM? So translational research is a journey or process which bridges the gap between the lab and society. Translational medicine is clinical application which bridge the gap between lab and patient. And EBM or evidence-based medicine is a decision-making framework which applies the best evidence in healthcare. So this is a takeaway for you that translational research is journey, translational medicine is into clinic, and EBM is a compass or a framework. So remember one thing, any research is of little value unless it reaches the patient and community. And therefore, translational research is very, very important. So thank you. And I have taken only 15 minutes more.
Not at all, madam. It was a really very excellent speech we listened today. Participants, if they have any question, they can post in the chat box.
Yes, Vinay Pawar has said that translational research has to be a departmental vision, very rightly said, because every time a new student comes to department, we give a new topic to that. If you remember what I have said that Dr. Dahanukar, who was my mentor, in her department for 20 years, every student was working only on Tinospora cordifolia. So there was no choice. Means the students used to understand that if I am going to get the Hanukar madam as my guide, I have to work on Tinospora cordifolia only. Now it has happened to my lab also that if I am posted in this lab, I have to work on Trifle. There is no option. So yes, this is a departmental mission and we have to have these kind of departmental missions in Ayurveda colleges also. So we can generate some data in 5 years, 7 years course of time. Do we really use Triphala Phalamajja in medicine? Actually, when we made it prepared, we have used Phalamajja. So ideally, Phalamajja should be used. This is for organizers that please make, and it has been already given. So yes, there are no more questions.
There is a question.
Yeah, can you elaborate regarding the Prakruti predicting the drug response? So this is like in modern medicine, it is like slow metabolizer, fast metabolizer. So in our case also, Pitta Prakriti, if we say that it metabolizes anything, drug, diet, fast, then maybe we have to keep like the treatment schedule quite often. And in Kapha Prakriti, maybe delayed response, so the drug will be released in body slowly, and we have to, I mean, it can be like the dosage schedule can be different. If you read the paper which is published by me, like that is a Prakriti and response to platelet function, platelet aggregation and Prakriti, and response to aspirin. So there we have shown that how in Pitta Prakriti the situation is very tricky, because in Pitta Prakriti the response to aspirin is poor, and we have to use higher doses in Pitta Prakriti. And if we use higher doses, then there will be bleeding chances, because aspirin is known to cause blood thinning. There will be high chances of blood bleeding, and in Pitta Prakriti this risk will be more pronounced. So how fragile this condition is, that if you have to manage Pitta Prakriti individuals with aspirin, the condition is very fragile. So likewise, I was discussing about the drug predicting responses.
I think there are no further questions. Okay. So, ma'am, so Dr. Supriya, if you have gone
Yes, sir.
through the curriculum, there are few practical parts and experiential learning part of it. So, could you just guide us on that? What practicals can be done by these,
So
especially?
we can have like we can create some cases for evidence-based medicine. I think there is a experiential learning, so we can develop some cases. And like now the situation is changing, and I know when you are in the chair, I mean in the like main driving seat, Mohan sir, the situation will change further. But actually, when we were studying how the questions paper used to be there, like write a ama chikitsa, ama nidan, so it doesn't actually uses EBM anywhere. So if we can create some cases, and that case-based learning, what you call, I'm not expert of medical education technology. You can, I mean, frame it in a better way. But these kind of evidence-based, so then the students can go and search for the references, that whether any clinical study, either any case report is published on this topic, and then using that evidence, what treatment can be planned for a patient which is mentioned in the case. That is something which we can do.
Absolutely. Any more questions? No. Yes, thank you, Dr. Supriya ma'am. What you said is absolutely right. So this particular part, the entire seventh module itself is very much challenging for everyone, because we have to find many resources, generate resources, either search for them or create them. So creating them requires the thorough understanding, and probably this lecture series will help everyone to go to the depth of that understanding, and that can work to generate the resources. So it may be the finding resources in the form of the papers, or generating some simulated cases or evidence based cases, or themes or scenarios, developing some of the scenarios which can be used as the uh, models for conducting the practicals. And then student may find that kind of things, availability of those things in the research areas as well as journals, and that can work as experiential learning. So probably uh, this was a very tough task to frame practicals and experiential learning sessions on entirely literary part. So still they are placed, and probably we are going to explore some of them in the coming lectures on Monday and Tuesday. So thank you Supriya ma'am. Over to, I think we can conclude the session.
Yeah, thank
Vikram
you.
sir, just summarize the session and conclude.
Okay, thank you, Dr. Mohan sir. Thank you for this brainstorming session, Dr. Supriya Bhalerao ma'am. Going from the fundamentals, big picture, its various impacts, importance of publication, and go beyond publication too. Importance of translational research, their key areas, challenges, the real one, and EBM, evidence-based medicine, and its 5-step model. Ma'am has explained everything today. Thank you, ma'am. Once again, thank you, ma'am. Also, I took this opportunity to thank Dr. Aishwarya ma'am and Dr. Sheetal ma'am for sharing their research with us. Thank you, one and all, for the participants for their patience listening. Thank you, ma'am.